Case 9 is of an 85-year-old woman who resides in a nursing home. She was treated for urine and blood infections for a week, and then she was taken to the hospital as she developed copious diarrhea. Stool, midstream urine, and blood samples were collected. The stool sample contained moderate numbers of red blood cells and white blood cells. However, no pathogen was identified from this sample. The midstream urine showed low numbers of red blood cells and moderate amounts of white blood cells and epithelial cells. Similarly to the stool, no significant growth was identified from the urine. The blood sample had no growth 5 days post-culturing. The lack of pathogen identified from the stool sample is suggestive of a normal coliform being responsible for the woman's symptoms. Normal gut bacteria can become pathogenic under some conditions, such as antibiotic intake. The presence of red blood cells and white blood cells in urine is indicative of an ongoing infection. However, no pathogen was identified in this sample either. This further supports the theory that a coliform has caused the infection and its presence could be masked by contamination of the urine, as indicated by the elevated epithelial cell count. The lack of bacteria in the blood could be because that infection has resolved. As the woman was taken to hospital due to copious diarrhea, we believe the stool sample is most clinically significant and is therefore our primary focus. There are many unknowns in our case study, largely the patient's history and the lab tests conducted on the samples. It is beneficial to know the medication plan the patient was on, at what dosage she was ingesting them, and how often. Additionally, the duration of her infections needs to be acknowledged because depending on the causative agent of the infections, there could be long-lasting repercussions which can change the patient's normal flora and the types of cells present in specific areas of the body. As the urine sample is contaminated, repeating the urine test with a new sample would be beneficial as it would show accurate cell populations whose ratio can be indicative of the presence of disease states. Furthermore, it is unknown what tests were conducted on the 3 samples. Therefore, we are running under the assumption that they were tested and cultured under normal laboratory sample procedures. Several presumptive organisms were considered, including E. coli and C. jejuni. However, these bacteria were eliminated due to inconsistencies between their characteristics and the patient's clinical presentation. C. difficile was also considered, as it fits the criteria of causing diarrhea, invoking a pro-inflammatory immune response as seen by the number of white blood cells in the stool sample, posing a greater risk to the elderly and being more common in people who frequently stay in hospitals or long-term care facilities such as nursing homes. C. difficile is also strongly linked with antibiotic-associated diarrhea. Some strains are toxicogenic and can cause inflammation of the colon, which may result in bloody diarrhea.
Direct examinations of samples were then completed to observe for any immediate abnormalities. This refers to any wet mount preparations, immunological and genetic testing, which may assist in a more immediate identification. Additional examinations could have been an ova and a parasite exam. All 3 samples should have been incubated by inoculating fecal matter on both selective and differential media at the discretion of the lab to observe and collect isolated colonies. Typically, stool samples are plated on horse blood agar, anaerobic horse blood agar, McConkey XLD, CAMP, and CIN. For urine samples, these plates should be inoculated: horse blood agar, McConkey, and chrome agar. The blood culture was analyzed using BACTEC and BACT Alert automated systems and showed no growth after 5 days. The case study showed no blood infection and no significant growth for both midstream urine and stool cultures. Therefore, the negative culture shows it could be normal flora of the gastrointestinal tract causing the symptoms, and that the isolation of C. difficile is needed to further distinguish the causative agent. Then the Remel Clostridium difficile agar and Chrome ID agar are required for organism detection and isolation. The flat circular colonies with filamentous edges that demonstrate a yellow zone extending 2 to 3 millimeters from the edge of the colony should be collected after 48 to 72 hours of incubation at 33 to 37 degrees Celsius in a conventional anaerobic gas jar. To confirm that anaerobic conditions, the C. difficile colonies will be subcultured to a blood agar plate and inoculated at 33 to 37 degrees in ambient air. The Chrome ID agar will be incubated in anaerobic conditions at 37 degrees for 24 hours, and the gray to black irregular or smooth bordered raised opaque colonies will be collected. The presumptively identified organism is Clostridium difficile. Clostridium difficile is a gram-positive, anaerobic, spore-forming, toxin-producing bacillus. As this bacteria is anaerobic, it is well suited for the environment of the gastrointestinal tract, especially the colon. Once the bacteria reaches the human intestine, the spores germinate into their disease-causing vegetative state with the presence of glycine and cholate derivatives. Large numbers of C. difficile in the colon result in the production of its two exotoxins, toxins A and B, and these toxins produce the pathogenic effect of the bacteria. The toxins cause cytopathic effects through toxin A inactivation of RAP proteins, which are responsible for the regulation of cell-to-cell junctions, which leads to the disruption of intestinal epithelial integrity.
The cytopathic effects of the toxins A and B are drastic morphological changes such as shrinking and rounding of cells. This is initially accompanied by a formation of neurite-like retraction fibers, which block the Rho-dependent signaling interfering with the actin cytoskeleton and disruption of the tight and adherent junctions, which leads to loss of cell-to-cell contact. This increases epithelial permeability, which then results in diarrhea. The C. diff toxins can also induce cytotoxic effects on the intoxicated cells. This is associated with the activation of the inflammasomes by the glycosylated Rho-A, which can then cause inflammation of the colon, which is also known as pseudomembranous colitis. Glutamate dehydrogenase enzyme immunoassay tests for the glutamate dehydrogenase, which is a metabolic enzyme found in all ribotypes of C. diff. This test is used to identify a large number of the bacteria in the gastrointestinal tract. However, it cannot be used as a standalone test to confirm the presence of toxigenic strains. Toxins A and B by enzyme immunoassay are used as the rapid method for detection of C. diff. This method tests for the presence of the toxins. The results, however, need to be compared to that of the GDH results, as there is not only a positive or negative, but it may be equivocal. The use of light cycler PCR assay allows for the detection of C. diff and the toxin B gene. DNA is directly extracted from stool specimens and C. diff. 16S DNA and toxin B DNA are amplified on light cycler real-time PCR platforms. The identity of the sequence is confirmed by monitoring the binding of specific fluorescent probes to each of the amplicons and subsequent melting point analysis.
As a causative agent was identified as C. difficile, antibiotic sensitivity was only tested on the antibiotics used to treat this infection. The inherent problem of C. difficile infections is that they are resistant to virus antibiotics, therefore allowing for proliferation and toxin production. So it is imperative that the antibiotic treatment is both effective against C. difficile and permissible to normal flora of the gastrointestinal tract. C. difficile is mostly sensitive to metronidazole, vancomycin, and fidaxomicin, and this is clearly seen by the size of the zone of inhibition. Metronidazole is usually the antibiotic used against anaerobes. Vancomycin is a glycopeptide antibiotic that works by killing the bacteria. Fidaxomicin is a new antibiotic that inhibits C. difficile. Additionally, a million inhibitory concentration test of metronidazole needs to be performed as this impacts at what dosage the patient takes the medication. The method currently recommended by the Clinical and Laboratory Standard Institute for C. difficile is an agar dilution method using Brassilia agar supplemented with hemine, vitamin K, and 5% lactate blood. A susceptible reading of metronidazole for Cidofusel is an MIC of less than 8 microgram per milliliter. The glutamate dehydrogenase assay will be completed in the laboratory and should show a positive result. Then the toxin A and B test will be used, and there is a high possibility of getting a positive result. However, the equivocal or negative result could be presented. For the equivocal or negative result, the molecular detection of toxin genes is required. For this patient, the result should be positive for toxin A and B test or indeterminate toxin A and B test result, but the molecular detection should consistent with Cidofusel. To determine the treatment, the antibiotics that patients use for urinary tract infection and blood infection should be stopped. The diarrhea symptoms usually stop within 10 to 12 days. If the symptoms still present, the antibiotics effective against Cidofusel, such as oral metronidazole, vancomycin, and fideoxomycin, should be used. Thanks for listening our presentation, and this is our reference list.